Okay, good morning, everyone. So first of all, thanks for the organizers for inviting me and for the opportunity, of course, to share some snapshots of some of the work we are doing in the lab. Okay, so today I'm going to talk about, my talk, I want to highlight something that was also highlighted in the previous talks about bioinformatics. So bioinformatics is not about analyzing data or integrating or developing methods for data integration or analysis. It's more about taking hypotheses from very early stages, basic research perspective through all the steps all the way to clinical application, whether it's for developing a vaccine or a treatment or a biomarker for a disease. So, and this is exactly what my talk is about today. And I will be talking about one of the projects we are conducting in the lab about long COVID. So what is long COVID? I guess you are all familiar now with COVID-19, which, generated or created, a worldwide health issue with millions of deaths, and fortunately there are vaccines, but unfortunately a fraction of individuals who got COVID-19 developed what we call long COVID, and long COVID now is becoming a disease. It's an inflammatory disease, maybe a metabolic related disease, maybe something else. We don't know yet exactly what long COVID is about. All what we know for sure is that fraction of people who got COVID-19 has developed some type of symptoms that ranges from general fatigue, intolerance to exercise, and many other more serious conditions like brain fog, memory loss, among many others. So the statistics of long COVID, as estimated, I think as of 2023, we kind of estimate the long COVID cases worldwide to 30%, anything between 20, 30, 40, depending on what website you are looking at, of COVID-infected individuals. Just imagine that 30%, or let's say around 30% of individuals has a condition called long COVID, for which we don't have any vaccine or treatment so far. So there was a treatment, yeah, sure. So there was, I believe there was a treatment, Paxlovid, that was tested 1 year or 2 years ago, but it didn't protect from long COVID, or it didn't resolve. So back to my talk, the demographics of individual who have long COVID is not specific, so there is no clear or significant association between gender, age, or even ethnicity associated with long COVID. But there is a fact that people with underlying health condition or chronic diseases are more susceptible to develop long COVID compared to the ones to healthy population. Okay, so what are the underlying pathogenesis of long COVID? So one of the underlying pathogenesis of long COVID is persistent viral of the virus. So after COVID infection, in most cases, if not in all cases, the virus is cleared in the blood after 2 to 3 weeks, depending on the individuals, of course. But let's say there is a 2 weeks period where the virus peaks in the blood, then it's cleared. But the virus persists in tissues. So the virus is cleared from the blood. In some patients, it persists in many tissues. And this is one of the reasons we think could be leading to long COVID later on. Latent viral reactivation. It was shown clearly now by many labs, including the lab of Akiku, that COVID-19 is involved in reactivating dormant viruses, such as EBV, among others. Okay, so in some people, we all have dormant viruses like EBV and others. In some people, actually, when they get COVID-19, this leads to reactivating some of these viruses because of some type of immune environment that is that favors this immune reactivation or this viral reactivation. Persistent inflammation, this is a hallmark of long COVID. So long COVID patients in general, all of them have persistent or chronic inflammation years, 1 to 3 years later after COVID infection. And autoimmunity, was shown also that SARS-CoV-2 or COVID-19 triggers autoimmune disease. Okay, so what did we do? We were able to access different cohorts of patients, a control cohort from before the COVID era, from 2014, I believe. So we were kind of certain that this individual do not and never had COVID-19, hopefully. We have also a cohort of COVID-infected individuals where the infection was resolved. So the infection was cleared and these individuals are doing great. We also collected samples from a long COVID cohort. These are individuals who had COVID-19 but showed symptoms of long COVID 3, 6 to 1 year after infection, after initial infection. So the table here shows the demographics and the number of patients within each group. So you have the negative controls, the convalescent controls, which are the CC, the long COVID, which are the LCs, and there is another group of acute COVID-19 infection. These are individuals from which we collected samples during the acute phase, during, let's say, the 1 to 3 weeks after infection, okay, less than a month for sure. Okay, so what we collected different type of samples for different type of assays. We collected PBMCs or whole blood cells for transcriptomics profiling using bulk RNA sequencing. That's what we did. We didn't do single cell for this project. We collected plasma for proteomics profiling, and for proteomics we use this platform called SomaScan that screens for 11,000 proteins, human proteins. And we also conducted a couple of other assays like looking at the virus, different strain of COVID-19. We did neutralization assays for different also strains, and we looked at cellular responses, looking into interferon gamma ELISpot responses. And the bar plots on my slide shows the prevalence of some symptoms in the long COVID patients. So as you can see, among the symptoms, you can see that shortness of breath, fatigue, and other issues or health-related issues are the most represented in the long COVID cohort. But the issue here is that when you look at the list of symptoms, they are not specific to long COVID, right? These are symptoms you can get when you are weak, when you have a cold, when you have any other viral infection. So based only on the symptoms, we cannot really say this person has long COVID and why compared to someone else who is just getting sick. Okay, so this is the study design. We collected cells and plasma at different time points from all 4 groups. The acute phase, or the first weeks, which is less than a month, 3 to 6 months, and in some patients we were able to access samples after 1 year, after the initial 1 year after initial infection. So this is just a schematic showing that in blue, in long COVID patients, there is persistent immune activation through 1 year. However, in the convalescent controls or the CC, the people who did not develop or transition to long COVID, the infection was resolved after a month or so. Okay, so first results is from the viral screening. So we screened for different strains, as you can see on my slide. The first one, the Wuhan 2022 or Washington 22, the Delta and the BA1. And we compared the CCs, which are the convalescent controls, to the LCs. And as you can see on my slides, there is little difference, if not significant at all, between these 2 groups. And remember, this is blood. We measured the virus in the blood, not in tissues. We couldn't access the tissues from these individuals, obviously. So when we look at the blood, just to summarize this slide, there is no differences when we looked at the neutralization titers, and the virus as measured by subgenomic sequencing. But we do see a small difference, higher T-cell responses in the long COVID group as shown in panel B compared to the CCs. Some of it is significant with a cutoff of 0.5. So we do we hypothesize that the higher cellular immune responses in the LC or the long COVID group may reflect more severe acute infection or persistent virus in tissues that is maintaining this, highly activated immune environment leading to high cellular responses. Okay, back to the transcriptomics now. So we focused on the period between 3 to 6 months because this is the range, the time frame where we have enough samples to get statistical power and significance. So we performed bulk RNA sequencing in all groups from the period of 3 to 6 months. And this is the first plot is a k-mean analysis, mostly a PCA, showing the overall distribution of the transcriptomic profile of the 4 groups compared to each other. So clearly you can see that the long COVID group shown in blue is different from the remaining 3 other groups. And you can also see that in acute infection, that is shown in purple, is kind of closer to the convalescent controls and to the uninfected individuals. They are not the same, of course. There are still differences between the 3 other groups, but the main conclusion from this first unsupervised clustering or profiling is that during the period of 3 to 6 months, long COVID patients have something different from the convalescent controls, from the acute infection, and even compared to negative controls. And when we looked at briefly to some of these differences, we do see that markers of inflammation, immune activation are the ones that are highly increased in the long COVID group compared to the other groups. And here I'm showing the comparison to the CCs only and the negative controls as shown here on the first slide on panel A. And so it's clear that LCs or long COVID patients have more inflammation, persistent immune activation, 3 to 6 months after initial infection compared to the negative controls and the convalescent controls. Now, on the other hand, if you look at panel C shows markers of T cells, cytotoxic T cells, and NK. For some reason, which I'm going to come back to later on in my talk, markers associated with NK, natural killer cells, and CD8 T cell are less activated or downregulated, if I may say, in the long COVID group compared to the CCs or the negative controls. Now, when transitioning from the genes into pathway analysis, and this was done using gene set enrichment analysis, we in the first panel, I'm just showing a scatter plot of the major genes upregulated in red or downregulated in blue in the LC compared to the CC groups. And the remaining plots show the pathway analysis of all signatures that were screened for that showed significant increase in the long COVID compared to the convalescent controlled individuals. So among these pathways, we kind of compiled them into modules. So you have a module of cytokine, pro-inflammatory cytokine signaling, which is perfectly consistent with what is known about long COVID, persistent inflammation, including persistent activation of cytokines such as IL-6, IL-1, and TNF, etc. The second module is complement activation. And if you are familiar with COVID-19 literature, you may know that the complement activation is one of the hallmarks of COVID infection, leading to coagulation and other issues in in severe COVID cases. The third module is immune cell signatures. And briefly, if you look at the names, most of it are monocytes and macrophages, pro-inflammatory immune cells, suggesting that, consistent with the gene analysis, of course, that signatures associated with immune activation, inflammation are highly increased are persistently increased 3 to 6 months in the long COVID patients, Whereas in the CCs or the negative controls, or even in the general population, when you get a viral infection, everything is resolved within a week or two. 3 to 6 months, there is nothing left, right? There is no activation, no inflammation, except, of course, if the patient has different health issues. Okay, so in addition to that, we found that there is a module of metabolic-associated pathways that was also increased in the long COVID patient, suggesting that long COVID patients have also a metabolic shift. I'm not going to discuss the metabolism of long COVID patients today, but just know that in addition to inflammation, immune activation, there is a metabolic shift in long COVID patients compared to convalescent controls. And back to the downregulated pathways, and consistent with what I said before, pathways associated with CD8, NK, and DNA repair were downregulated or inhibited in the long COVID patient, suggesting that long COVID patients, in addition to inflammation, chronic immune activation, immune exhaustion, they also have issues with tissue repair or DNA repair. Okay, so this is just same back to the CD8 and the immune exhaustion. So we found that there is an increase of immune exhaustion leading to chronic, because of the chronic inflammation and the persistent immune activation. Of course, the immune system of this long COVID patient gets exhausted at the end. Okay, now how about after 6 months? We took the signatures that were highly increased in the long COVID patient at 6 months. We looked at after 6 months, from 6 months to 1 year, a little bit after 1 year, depending on the samples we had. And we found that not only these pathways were increased 3 to 6 months, they persist also after almost a year in the long COVID patient, suggesting that this persistent immune activation and chronic inflammation was found significant in the long COVID patient 1 year after initial infection. This is a network analysis just to highlight the major hubs or the major markers among the pathways that were increased in the long COVID patients, and showing JAK-STAT signaling, IL-6 receptor, among others. This was done using WGCNA, which I hope you are familiar with. If not, please feel free to ask a question. And this is our model for long COVID. And I'm happy to say that based on this analysis and this bioinformatic analysis, we were able to take this project from initial hypothesis into clinic. And now our lab is initiating a clinical trial in partnership with industry to develop a treatment, for long COVID, which is the JAK-STAT inhibitor, because JAK-STAT and IL-6 were among the top pathways that were found to be increased in the long COVID patient. So this is a clear case of how bioinformatics can lead to clinical applications or treatment development or vaccine development. Okay, I will end with my acknowledgement slide and happy to take your questions.
We slightly over time, so we will only take one burning question.
Yeah, what is the role of the cytokine storm? You mentioned interleukin-6 downregulation.
Upregulation in the long COVID patient. Yeah, so IL-6 was known to be highly induced in the after COVID-19 infection, and now we are showing that is associated with long COVID. IL-6 and IL-1 and other pro-inflammatory cytokines are, when there is a viral infection, there is a peak of these pro-inflammatory cytokines that should be resolved after a certain time. If it's not resolved, they will maintain a certain level of immune activation of activating other pro-inflammatory genes and immune cells, like monocytes and macrophages, maintaining this cycle of inflammation, leading to chronic inflammation and, worsening of disease.
So that means cytokine can be mapped in long COVID patients?
Yeah, totally.